Welcome to all. Um, Today, in this lecture, we are going to discuss history and scope of immunology. I'm Mahendran from the microbiology department. Let us see the structure. So, here in this video, we are going to deal what is immunology and what are the scopes under the immunology and what are the mechanisms behind for the particular immune system and what are the contributions given by the various scientists to develop the field of immunology. Immunology is the study of the immune system and its responses to invading pathogens. The physiological function of the immune system is to prevent infectious and to eradicate established infections. Immunity is defined as resistance to disease, specifically infectious disease. A collection of cells, tissues, and molecules that mediate resistance to infections is called the immune system. Let us see the scope of immunology. We are having various scope under the field of immunology. Let us discuss few scope under the field of immunology. Under the immunology thing, we are having the immunity. So in the immunity, we are having two major types, that is passive and active. So here the passive immunity is a naive individual receives cells and boosts the immune system. Passive immunity is therefore useful for the rapidly conferring the immunity even before the individual is able to mount an active response, but it does not induce long-lived resistance to the infection. And as well as the passive immunity will helpful to the particular individual for the production of long-term, not a lifetime production for the particular host. They are having different kind of infections, so definitely they should take in between their remedies or treatment for the particular disease. Okay, next, non-self substances from the immunological viewpoint, but drugs and even normal food, etc., which are of course non-self too, can sometimes give rise to immunity. Detection of non-self material is carried out by range of receptor molecules, infection, parasitic viruses, bacteria, protozoa, worm or fungi that attempt to gain access to the body or its surfaces are probably the chief of the immune system. Higher animals whose immune system is damaged or defense succumb to the infections that normal animal overcome. Adaptive immune response, the developmental augmentation of defense, it can result in elimination of the microorganisms and recovery from the disease, and often leaves the host with specific memory, enabling it to respond more effectively on a reinfection with the same microorganism, a condition called acquired resistance, because the process by which the body puts together the receptors of the adaptive immune system is random adaptive immunity, sometimes responds to harmless foreign material such as the relatively autoimmunity. Vaccination is a method of stimulating the adaptive immune response and generating the memory and acquire resistance without suffering the full effects of the disease. So the vaccination has different types: attenuated, killed, live vaccines. Attenuated vaccine is nothing but the particular gene or material alone we have to remove or take it out to make it as a vaccine or drug. And killed vaccine, only we have to kill the whole microorganisms we can use to as a vaccine to treat the particular disease or preventing the particular disease. And one more thing is, live vaccines, we no need to do anything, directly we can take it as a the whole microbes to make it as a vaccine and give it to the particular disease against for the particular host for the different kind of disease. Okay, And one more thing is vaccination should be in the particular level, and the doses should be needed. And if we need for the further production, we have to take it as a booster dose. The names comes from the vaccinia or cowpox used by Jenner to protect against smallpox. Grafting cells are organs from the another individual usually survive innate resistance mechanisms but are attacked by the adaptive immune response leading to rejection. Autoimmunity is normally stimulate the adaptive immune responses because of a variety of special mechanisms that ensure a state of self-tolerance, but in certain circumstances they do not stimulate a response, and structures are foreign or condition is called as autoimmunity or automaticis. So different kind of autoimmune disease or disorders are there. So it should be taken care and attention to the previously, otherwise it leads to severe problem to the particular host or individual. Next, hypersensitivity. Sometimes the result of specific memory is that to re-expose to the same stimulus as well as or instead of eliminating the stimulus as unpleasant or damaging tissues. This is called hypersensitivity. Example are allergies such as hay fever and some forms of kidney disease. Immunosuppression, autoimmunity, hypersensitivity, and above all, and graft rejection sometimes will necessitate the suppression of adaptive immune responses by drugs or other means. The hypersensitivity has been classified into 4 different types. Each type of hypersensitivity will cause the infection to the particular host for the different types of affecting their immune mechanism for the particular host system. Next, we'll see the what are the persons who are giving their contribution to develop the field of immunology. So here we are discussing only few scientists because they are the major, they leads major contribution to develop the field of immunology. So we'll see the only the few scientists for the particular development of immunological field and with their contribution. First, we will see Edward Jenner. Edward Jenner carried out a famous experiment on a healthy 8-year-old boy, James Phipps, with cowpox. Successfully, he identified he taken out the lesions of the cowpox lesion from the particular boy, James Phipps body, and he made it as the vaccine, and he converted the different form and transferred to treating the particular cowpox disease. He took a material from the both person on the arm and same names who had apparently contracted the cowpox. So in this experiment, he discovered the vaccine for the cowpox. So using with the person James Phipps. Next, we'll see Louis Pasteur. He prepared attenuated vaccines for the cholera, for the microorganism caused by Vibrio cholerae, and anthrax causing by Bacillus anthrax, and rabies. Uh, these are the vaccines discovered by the Louis Pasteur. It is one of the major milestone to develop the field of immunology. Next, we'll see the person Elie Michnikov. He discovered the basis of leukocyte phagocytosis. Phagocytosis is nothing but it's a process to engulf the pathogens and eliminate it. So it covers the series of process like once the macrophages will engulf the foreign particle or microorganisms or other thing unwanted material and will take it to the phagosome, and then phagosome will attach with the lysosome, it converted as a phagolysosome. In this phagolysosome, here it will be producing some kind of enzyme. That lysosome enzyme will excrete or uh, convert the particular product or foreign material or microorganisms, it will be converted in the simplest form and it will be inactivated by the lysosome, and after it will be eliminated by as a waste product. Okay, so this is the process we'll discover under the contribution done by the Eli Mechnikov. Next, he discovered the cellular immunology. So this cellular immunology only will deals the immunity of the cellular mediator, and same way it will resembles and sometimes it will work out for the humoral mediated immune response. Next person is Paul Ehrlich. He developed the series of tissue staining dyes, including tubercle baseless. Nowadays, it's an important technique to staining the particular tissues or whatever may be the microorganisms. So it will be major one to staining the particular tissues to useful for the different identification or classification of the particular microorganisms. He discovered the staining technique using the staining method for the identification of tubercle bacilli. Worked with the coach. He also worked with Robert Koch and developed the antitoxin and hemolysis work. And antitoxin will be developed for the microorganisms or in disease as a diphtheria. Thank you.